right, so let's go through the process of protein synthesis. Let's first start out with a strand of DNA. So here we've got a strand of DNA, and we're just going to do 1/2 of it. So let's do ATC, CAG, GAC. So this is just 1/2 of DNA because a strand of mRNA only is copied off of 1/2 of the strand of DNA. So there's still the double strand of DNA, so on and so forth. But we're just going to focus on this one strand right here. So the first thing that's going to happen is we're going to make a strand of mRNA from that strand of DNA. So we've got our mRNA. So remember, in mRNA, T does not exist. So here, A is going to base pair with what? A is going to base pair with U. So that means T is going to base pair with what? T is going to base pair with A, and C is still going to base pair with G. Okay. C is still going to base pair with G. A, again, is going to base pair with U. G is going to base pair with C. G again with C. A with U. And C with G. Okay, so that right there is the process of transcription. And because it is transcribing the DNA, DNA is found in the nucleus, so this means that this process happens in the nucleus. All right, so let's kind of go back and let's talk about some terminology. So here in our DNA, each group of 3 is going to be a what? So each group of 3 in DNA is called a triplet. And each group of 3 in mRNA each of these are going to be called a codon. Okay, and so if this was happening in real life, that strand of mRNA would leave the nucleus and it would go find a ribosome either in the rough endoplasmic reticulum or in the cytoplasm. So let's say that this mRNA has found itself a ribosome. And there's actually 2 little spots to the ribosome. And let's go ahead and rewrite this. So UAG, GUC, CUG. So again, this is mRNA. All right, so the next part of this is translation. Let's go with green. Okay, so the next part of this is translation, and we're going to then, so a tRNA is going to bring down a specific amino acid based on the codon. So here, these spots right here, that is going to be the anti-codon that's attached to or is part of the entire structure here, which is your tRNA. Okay, so let's do the anticodons. So U is going to base pair with A with U, G with C. G is going to base pair with C, U with A, C with G. So C is still going to base pair with G, U with A, G with C. So those are your anticodons. But here, what we're going to actually use to determine the amino acid is your mRNA codon chart. So we're using our mRNA to determine what the codon is. So let's look at this. So we're going to go from left to right. So we're going to go this direction, just like reading a sentence. And here it says the first base. So this is the first, second base. This is the second. And then the third base. This is the third. So let's look at this. So we've got U, which is here. Second base we've got, which is an A. And then we've got G. So this is the U area here. This is the A. And then we come over here and I started with a stop. So we're going to actually write in, this is a stop codon. And it does what it sounds like. It stops the protein. So there's a start codon that starts the protein. There's a stop codon that stops the protein. I just happened to pick the code that started with the stop. I'm pretty good at that. All right, so let's do this again for the next codon. So then we've got G. Here is G. Then we've got U. And we've got our C. So here again, we've got our G. We've got our U, we've got our C, so we're going to come over, and our next one is valine. So we're just going to use the first three letters, V-A-L. All right, then the next one, we're going to do the exact same thing. Let me get rid of this. So we've got C-U-G, we've got a C here, a U here, and the G here. So here's our C, here's our U, and then we're going to bring this over, and it's going to be leucine, L-E-U. And that's all there is to it. So here we have our DNA, which is our triplets, and from there, in transcription, we're going to make a strand of mRNA from that strand of DNA. And those groupings of three are called codons. That happens in the nucleus. That strand of mRNA leaves the nucleus, finds the ribosome, and what happens next is called translation. And that's going to happen at a ribosome. Okay, and then the anticodons match up with the codons, bringing that amino acid down. And so we know specifically that there's a stop amino acid, a val amino acid, and an LEU, a leucine amino acid. And that's what's going to then connect together and eventually make a nice protein. So a whole bunch of amino acids make up a protein. The bond that holds them together is a polypeptide bond.